Hello and welcome to our new topic of discussion, that is newer drug development. Now this drug development process is not that simple. It's extremely lengthy procedure and lots of monies are involved. So developed countries and big pharmaceutical companies who are having that much of money, they can only afford of that new drug development thing. So it's a tedious process and it doesn't guarantee that going through those tedious process you're always getting the positive results. There are still chances of failure of this drug development process, and lots of money, time and resources gets wasted. So it's a very lengthy process. Why? Because it involves different steps. It involves, for an example, isolation of a compound or synthesis of some new compound. It takes 1 or 2 years. Then it goes into the preclinical studies, that studies into the animals or animal experimentation studies, where again a different kind of studies been undertaken, which takes around 2 to 4 years. Then the permission to introduce that drug into the human subjects, that takes some time. And formation of a pharmaceutical product for introduction of that drug, again standardization of the doses, all these things takes time of around half to 1 year. Then eventual clinical trials, that involves phase 1, 2 and 3, and long term animal toxicity studies, so that takes tremendous amount of time, around 3 to 10 years. Then the permissions for marketing the drug, it takes a lot of time, like half a year to 2 years. And eventually, even after marketing, you need to keep watch on those drugs for safety or the long-term side effects. And these are phase 4 studies or post-marketing surveillance. So it's a tremendous tedious process. Around 10 years or 12 years gets involved in this whole process, and it doesn't guarantee that you will get always a positive results or a successful pharmaceutical product. So let's talk about different steps in brief, how the drugs been produced and what different stages it goes. So let's first discuss about what are the different approaches of drug discovery or invention. What are the different ways by which you can identify a molecule. So these are different ways or approaches for drug discovery or inventions. So drug may get discovered by exploration of natural sources. For an example, and we have a lot of examples that drug gets produced from plants. Like for an example, from poppy seeds or opium, morphine is been produced. Ephedrine will produce in from ephedra. Cinchona, anti-malarial drug, quinine is been produced from cinchona bark. Another anti-malarial artemisinin derivatives produced from quingasu. Belladonna, atropine is been produced, and D-tubercularine for curar poison. So all these are examples of natural sources and especially plant origin. So all these drugs been derived from plant source. We also have another source of natural aspect, that is animal sources. So many are drugs like adrenaline, few hormones like thyroxine and insulin now developed from animals, liver extracts and even NTCRs. So these are some of the examples of animal sources. And the breakthrough discovery of penicillin, that gave us one more source of drug discovery. In these are microorganisms. Drugs can also be produced from microorganisms. So this is how you can explore the natural sources and from which you can identify molecule of a drug. Sometimes drugs get discovered serendipitously. What is serendipity? Serendipity means by chance. Okay, so drugs, there are many examples of these serendipitous discoveries, and few of them are barbiturates and chlorpromazine. These are the drugs that serendipitously discovered, means scientists were working on some another project on another drugs, and by chance some new drugs they found out, some new activities they found out, and from there a new molecule was discovered, and that is called as serendipitous discovery of drugs. One more way of drug discovery is lead optimizations. Now, what is lead? Lead means any chemical congener, or sorry, any drug or chemical product which already having its pharmacological activity. Now what you want is you want lead optimization. So you want to optimize that lead. So you want to produce a new product from that known pharmacological activity, which are having more specific actions, which are having less side effects, or which are having high efficacy. So synthesizing a new chemical congeners of the compounds which are having no pharmacological activity for more selective actions or superior activity. So from known product, you are making one more product which are having better activity, lesser side effect than that known product. That is called as lead optimizations, and many of the drugs nowadays been produced by these concepts only. For an example, from amphetamine they produce methamphetamine, right? So they make some chemical changes so that drug becomes more efficacious, less disadvantageous. Yeah. So this is how the lead optimization works. Examples are thiazide diuretics, they derived from acetazolamide. Tricyclic antidepressants derived from phenothiazine antipsychotic derivatives. So there are many examples, and most of the drugs developed by this concept that is called as lead optimization. Now, one more aspect is important in this drug discovery is enantiomers. Now, what is it? Drugs, many of the drugs are chiral compounds or chiral compounds, right? What is it? It means it's having two compounds. We call it as enantiomers. So, these chiral compounds having two compounds, for an example, R and S, or D and L, so dextro and levo. So all these things we will see in particular chapters, that these individual compounds are called as single enantiomers. So R enantiomer and S enantiomer gets combined, and they are having this is called a racemic mixture. This is called a racemic mixture. So it's a racemic mixture of R enantiomer and S enantiomer, for an example. And these are called chiral compounds, which are having different enantiomers, and each enantiomer having individual activities, right now these enantiomers are combined, so you are seeing a combined activity. But if you extract that compound and if you introduce that single enantiomer, it will produce its peculiar activity. Yes, each enantiomer having its own activity, and combination having a different activities, and taking advantage of that, we have produced single enantiomers. For an example, many drugs are available as a combinations. So as a combination, they produces these different actions, and now we are extracting this enantiomer from it, and from that we can get some different activities. So only one enantiomer and its activity we can produce, and we can help this in producing newer pharmacological products with better efficacy and lesser side effects. These are some of the advantages of that enantiomer activities. For an example, S enantiomer of atenolol is effective in half dose as atenolol. It's better tolerated. Same goes for metoprolol. Amlodipine, see, less side effects with S-amlodipine, that is less peripheral edema. So using those enantiomer also having less side effect. S-omeprazole having better oral bioavailability, so better pharmacokinetic data, more potency with S-pantoprazole. There are many examples which are using this concept of using single enantiomer rather than the racemic mixture for better efficacy and lesser side effects. One more approach of drug discovery is rational approach. Now, rational approach is basically more of related to pharmacodynamics. In pharmacodynamics, we have seen how drug produces its action, and that is its mechanism of actions, right? So, on which molecule or on which pathway this drug acts, or on which enzyme this drug acts. So, identifying that through physiological, biochemical, and pathological knowledge, and making a target specific drug, that is called as rational approach. For an example, the last pump in the HCl production in the stomach is the proton pump, that is also called as H plus K plus ATPase pump. that is the last pump for production of gastric acid secretions. And if we block that pump, the production of gastric acid decreases tremendously in stomach, and that can be helpful in patients of peptic ulcers. So we have produced drugs which inhibit this H plus K plus ATPase pump. Similarly, GP2B3A receptors are the receptors which are responsible for platelet adhesions. Okay, platelet binding with each other, and that forms the platelet plug. So taking advantage of that knowledge and making the drugs which inhibit these GP2B3A receptors helps for platelet function inhibitor, or making a drug as an antiplatelet molecule. So this is how a rational approach can help us making some new drugs. Now, some of the advanced approaches includes molecular modeling. Now we have access to to the different structure and activity of different proteins in the body, like receptors and enzymes. And depending upon that, we can also produce targeting drugs as well, or targeting compounds as well. So example of it is making selective COX-2 inhibitors. Initially we had drugs, but it was non-selective. It was inhibiting both the COX enzymes, COX-1 as well as COX-2. But with the help of molecular modeling, it's now possible to inhibit only one enzyme, and making drugs which selectively inhibits that enzyme, so that we can get specific benefits. So that is molecular modeling, one more way of producing newer drugs. Combinational chemistry, you combine in random manner different chemical combinations and produce many number of compounds, and then screen those compounds for the pharmacological activities, but higher chances of failure in this type of drug discovery. And the biotechnology, very promising, where drugs being produced by recombinant DNA technology, like insulin, interferon, human growth hormones, and many of the drugs we've been using these days because of this biotechnological approach of recombinant DNA technology. So these are different ways by which we can produce a new drug or the new compound or a new molecule. And now we have produced a new compound or the molecule. Now it's a time to introduce that into animals for experimentations. Okay, so after the new molecule has been found out, or the new molecule has been developed by different techniques, now it's time to introduce them into the animal for experimentations. And that are called as preclinical studies. So after synthesizing a prospective compound, it's been tested into the animals for different pharmacological profiles. Now there are different types of animals whose physiology matches with humans. So depending upon the pharmacological activity, animals are chosen. Mostly first activities been performed on rodents like mouse, rat, guinea pig, hamsters, and rabbit. And then the animals been performed on larger animals, performed on larger animals like cat, dog, and monkey. So this can be asked in your examinations, like which different kinds of animals are used for experimentations. So now testing all animals having different types, and first is screening tests. So screening tests are done to detect presence or absence of any particular pharmacodynamic activity. For an example, if drug is having anti-seizure activity or not, if the drug is having hypoglycemic activity or not. So major pharmacodynamic activity is being checked through those screening tests. Then a specific activity detection is being done through test on organs or cultures. So on cell cultures and isolated organs, specific activity is being tested. For an example, if drug is having vasodilator activity or not, any anti-secretory or secretory activity or not. So this kind of specific activities been tested through different organs, which is been isolated from animals and bacterial cultures. Then to test human disease or efficacy similar to the human disease, we need to make animal models. So animal models been made. For an example, diabetes animal models been made, or for an example, inflammatory animal models been made. So that what is it? It is actually uh, creating a disease in animals. So you are creating diabetes in animal by different techniques. You are creating inflammation in animals, and now you are testing anti-hypoglycemic activities or anti-inflammatory activities on those animals. So these are called as animal models, where disease been produced and the drugs which cures those disease been tested. So test on animal models been done, then those confirmatory tests and analogous activities been done. We have already tested specific activity. Now it's time for exposure of systemic pharmacology, because in animals, apart from the activity of interest, there are different pharmacological systems there and that can interfere with the actions. So it is important to know different systemic activities of the drug as well. So systemic pharmacological actions also been noted by different tests. Then the quantitative tests, which mainly deals with dose response relationship of that drug, its potency and efficacy with existing drugs are compared. Pharmacokinetic testing's been done, and then important is toxicological tests are done, which main important for safety. So these are the series of tests which are done on animals with different aims and objective. Now, which toxicity tests been done? In toxicity also there are different toxicity testings been done. Some of the examples are acute toxicity, where animals been given a state drug of dose for around 1 to 3 days and effects been noted. For an example, LD50, that 50% of animal died because of which dose. So all these things been carried out and the data get recorded. Then the subacute toxicity testing, where for more time, for an example, 2 to 12 weeks, the drug is been given and its toxicity is been noted and the data recorded, like which organ it produced damage, then those organ been examined under microscope, which other toxicity it developed in animals. So all these things been noted. Chronic toxicity, 6 to 12 months drugs been introduced into the animals, and again its side effects and the pharmacological effects been recorded. Specific reproductions and teratogenical effects testings been done for effects on spermatogenesis, any effect on ovulation, if drug produces or having any effects on fertility or developing fetus. It's very important thing, so it's been checked. For mutagenicity, ability of drug to interfere with any genetic changes, right? So if drug produces any genetic defects or changes, so it is important for that mutagenicity is been done. And carcinogenicity, where during the period of animals life the drugs been introduced and kept watch for on them for development of any tumor. So that kind of different toxicity testing been done on animals.